So SDS provides the negative charge to the proteins, and based on which it separates on the gel, which is made of the polyacrylamide polymers. And since it is negatively charged, it moves toward cathode in the electrophoresis unit. Basically, before to understand that how we can troubleshoot the SDS PAGE gel or protein separation through SDS PAGE, we have to see that there are three important steps involved in it. First is that the preparation of the sample. It means the sample protein preparation from whatever the initial material is, whether it's a cell line, from the cell line, or from any animal tissue, plant tissue, or microbes or anything. So the first thing is to get a protein, whether it's a membrane protein or it's a cytosolic protein. The second thing is, once you get a protein, you purify it. Once we have protein which is ready to be go on the gel, you have to denature it and you have to reduce it using beta-mercaptoethanol or DTT, and also by heating it for 5 or 10 minutes depending on the sample and requirement based on the standard for each and every lab. Once you get that, you have a linear structure with negatively charged proportion to polypeptide chain length proteins, peptides, which you load on the SDS PAGE and you run it in the electrophoresis unit. You will get a gel with the protein bands, right? So mostly three basic steps. One is the sample preparation. Second one is that reduction and the denaturation of the protein, and third one is to load on the SDS PAGE system to separate it. After that, once the electrophoresis is done, you have to stain it using Comesi or using whatever staining method you choose for, and then you can visualize your protein. So basically these three steps. Now, if you have done everything correctly, you will be able to see a perfect separation, protein separation in the SDS, as you can see here in this particular figure in the method number 5, where you can see a number of bands, protein bands on the gel. However, if you find if something has gone wrong during the process, it will show some sort of distortion in the gel. And to understand how you can troubleshoot this, you have to understand that there are 5 steps where something might have gone wrong, because of which you are getting a distorted gel. First is in the sample preparation step. Second one is when you have prepared the sample and when you are loading on the gel. Third one is during the time of electrophoresis. Fourth one is once you have done the electrophoresis and the gel is with you, and at the time of protein staining. And fifth and one, you can also say that as a result of 1, 2, 3, 4, you can see that whether the separation has done properly or not in the 5th step, right? So these are the 5 steps which I will be just going through all these 5 steps in the next 5 to 10 minutes. Okay. So first step is the sample preparation. In most of the cases, if we are working with the cell lines, or if we are working with a very standardized protocol on a very simple cells or simple tissue, we generally do not get much problem in this particular step. However, if you are using some acidic solutions for the purification or for the extraction, then in that case, the pH of the sample may be too acidic. How do you find that your sample pH is not correct? When you are mixing it with the loading dye, it will turn into yellow. So once it is turning into yellow, it means that the pH of the sample is too acidic. The solution is that you can maintain the pH by adding CRISPR unless it goes back to the blue color. In second, which is the most common case in the sample preparation, is the contamination of the biomolecules. Contamination by DNA or secondary metabolites, mostly in case of when you are working with this recalcitrant plants, where you have a lot of secondary metabolites in the form of polyphenols, which interferes into the protein run in the SDS PAGE. So generally the one-step denaturing extraction, which is most commonly for the animal tissues or the simple cell lines, may not be suitable for the plant material. And in here you have to perform phenol extraction. And after the phenol extraction, you can precipitate the protein using the ammonium acetate in methanol. And in this particular image, you can see that the method 1, 2, 3, and 4 may not be, the protein bands are not very clear, they are streaked and it is a smear. Whereas in the step 5, which is mostly done by phenol extraction method, which removes most of the polyphenols associated with the proteins, gives you a very clear band in the SDS page. So that is the first step. If the pH of the sample is too acidic, it will turn the loading dye into yellow, which needs to be taken care by maintaining the pH. Second is that if there is a contamination of biomolecules, mostly in case of plant tissues, if it is rich in polyphenols or other secondary metabolites, then in that case you may have to use phenol extraction method. Right? So that is the first step. Second is that once you have already done the sample preparation, you have the protein, you have purified it, or you have removed all the contamination associated with the protein, then you have to load it on the gel. In this particular step, there are three issues which are more or less generally coming into the bad quality of the gel image. First is that you may have the low density of sample or the sample plus buffer. It means that how will you find it is that the protein or the sample will not stay into the well. In that case, the thing what you need to troubleshoot is that you have to check whether your loading dye is having sufficient glycerol or sucrose or not, which provides the density to the sample to stay into the well. Second is that sometimes what happens is that you do the overloading of sample or underloading of the sample. What it means is that when you are loading the protein onto the gel, you do based on the protein estimation. In case if you have done some error in the calculation of the protein estimation, you may overload the sample or you may underload the sample. In that case, the protein bands will not be very clearly visible into the gel, and that you can solve by making by doing a good calculation of the protein estimation. Sometimes this problem also comes when you make the well, when you do not make the comb, put the comb properly to make the well. So in that case, you can also check if the well is made using the proper comb thickness of the well, and if the integrity of the well is good or not. Sometimes you can also face the issues that in the precipitated proteins you see a lot of contaminants in the form of some dissolved contaminants. So that you can recover by doing the centrifugation of the sample prior to loading on the gel. Some of the examples which I have showed here, if you are doing a lot of protein, if you are putting a lot of protein on the gel, then you will end up something like this. If you are putting very less protein, then you will not be able to see a very good band. It could be a very light band. And the third gel image is depicting a correct or a good quantity of the protein which you can put on the gel. The third problem comes mostly is during the run, during the electrophorese run. If the run time is too short or if the run time is too long, that may be because of the running buffer, which can be either too diluted or too concentrated. This can also lead to heating of the buffer, and that can actually, due to the overheat or due to the runtime error, you will get a diffused band. Also, when you are taking too much time in putting the samples into the lane and before to start the electrophorese unit, there could be some lateral diffusion that also needs to be taken care. You have to do the process quite fast. And by mistake, if you are running the, if you are opening the electrophorese box without complete run in the power on condition, that could be even dangerous, and then you will lead to a protein gel, something looks like this, a distorted gel. As I said, that protein staining is always done after the gel is run, and that is a very important step in order to really visualize your protein bands on the gel. If you are doing a lot of proteolysis, or if there is a contamination in the sample or load buffer or impurities, you may end up having uneven background stains. And in most of the cases, if you are not putting a proper agitation or staking during the staining and the destaining steps, then you end up something like this, where you will see a lot of background, which is uneven background, and your bands may not be visible. So combining 1, 2, and 3 and 4 steps, which are basically the steps in the SDS page, if the gel does not contain protein, that could mean that there are no bands observed. And if you have done correct staining procedure, then then the problem could be in your extraction method, and which needs to be redo the extraction. If the band's results are poor, it could be because of the impure reagent, overloading, incorrect buffer pH, issues with the running buffer, or the high temperature. And the solution could be to prepare the new buffer in the pure water. Do not overload the sample. It means that you have to do a particular, a correct protein estimation before to load on the gel. Check the pH of the buffer in the use, including the loading buffer as well as the running buffer, and reduce the electrophrosis temperature. So mostly because of overheating also, you will get a distorted band. If the sample is having a very high salt concentrations in the proteins, or if there is any bubble in the gel, or if there are insufficient reducing agent in the load buffer, it can distort the band and it can create a fuzzy band. So you need to do the desalting steps during the sample preparation. You need to repeat with a new gel. You have to check the loading dye. It means that you have to check whether you are doing the things properly or not in terms of chemicals. If there are protein aggregation, it means that it is stacking well into the stacking gel, but then as soon as it is entering into the resolving gel, it is not resolving properly. So that could be an issue because there could be some insoluble material which could lead to this problem. So that can be solved by centrifuging the sample prior to load, decrease the sample load amount, and try using continuous buffer system. If the sample are not sufficiently reduced, that also could be a problem, which I said in the beginning also, that if you are not using a correct amount of beta-mercaptoethanol or heating step for the reduction and denaturation of the protein, that could lead into the dimers or the double band in the gels. So that needs to be taken care by looking into the loading dye and ensure the reducing time. Some of the common bad image of the SDS page, which I have shown here. If you are getting a good quality band, it's perfect. But if you are not getting a good quality band, that could be either a smeared gel. It could be because of the unevenly poured acrylamide mixture or due to cross overloading of the protein. You will see that the protein separating in two different wells are not anymore separate. It has completely smeared and went from one well to another well. The same you can see in here on the lane number 3 and the lane number 4, that there is overloading of the protein. And as I said in the past also, that if you are not doing your protein estimation correct, then you may overload the sample, and that can lead to an gel something like this. If you have a high lipid content, mostly in case of membrane protein, then you may be able to see this lipid smearing into the, you can see in the lane number 4 in this particular lane, that there are high lipid content because it is a membrane protein, and it can cause the smear in the gel. If the gel is not good, if the concentration of the acrylamide are not perfect, then you will again get a smeared gel, and also because of the overloading of the gel. So that could be the main reason to get the or smeared gel. Sometimes you will get a streak gel, and that could be because of the electric effect. And because of the non-uniform concentration of protein in the gel, it could be the electric effect, which can lead to the streaking of the gel. Another important thing to consider is that if you are not overlaying the overlaying pouring the overlay solution after the resolving gel into the gel cast, then it will lead to a rough edge, and that could lead to the streaking of the gels. So this is also very important that after putting the after putting the separating gel, you have to keep a this overlay the first gel so that the surface is smooth and it is not rough. In the third image, you can see that there are light bands, and it could be again because of the two major reasons. One, it could be a wrong protein estimation, so you might have loaded a less quantity of protein. Second is that there could be some issues with the staining of the protein, so you need to check whether your staining is working perfectly or not. Third is that it is also possible that when you are loading the protein, it might have came out of the well because the wells are shallow and maybe the comb didn't work properly, and because of that you lost a lot of protein into the buffer. So also, I was thinking that it will go in a way that Aditya would show you how to do the how to do the mobility calculation, but nevertheless he is going to show you soon after this. If you are missing the die front in the first part, then that could lead to the wrong calculation or compromised calculation into the relative mobility. So that is also important to run the gel into for the perfect timing in order to get all the resolved proteins into marker proteins in the gel. Also, it is not advisable, or it is not good to stop the uh, run early, because then in that case you may not be able to see all the proteins resolved into the gel, as you can see in this particular example. If you are not making your acrylamide concentration, if it is too high, or if you are not making in a proper way, then what will happen is that smallest protein may enter into the gel, but it may not be able to resolve properly, as you can see in this dense gel. Same, there is this electric field effect, mostly because of the spacer which interrupts the electric field at the edge. So there is a pull and the push, if you prefer, that effect, which can mostly affect on the side wells. So the side well will tend to move little inside, and that could lead to this kind of electric field effects, pulling of the bands in the gel. Also, if you're doing a very slow loading, then that could lead to the lateral diffusion. If you are opening the gel box without power off, you will lead to this distorted gel. Again, as he correctly explained you about the importance of current and the voltage, if you are overheating the gel, then the proteins will be distorted, and the gel will look something like this. So in a summary, if you understand that the first step to get a good SDS page image or the protein separation is to do a correct protein extraction, and it has to be devoid of any contamination from mostly from the biomolecules in case of plants or the algae. That can So what you need to do is a purification. Sometime if there is a problem of the pH, you can maintain the pH by the tris buffer. Sample loading, it means that you have to look carefully whether your loading dye is having correct amount of beta-mercaptoethanol or DTT, or if you are hitting the samples properly, or it means conclusively it means that whether you are reducing or denaturing your protein in a proper way or not. Also, whether your well is correct or not into the gel, and whether you are using a particular perfect comb for the gel plate what you are using. It should not be thick or thin. That can lead to like going out of the well. Your proteins may go out of the well, and then you may not get a good gel. Also, it has to be not very overheated, because that can lead to a distorted gel. And finally, if you have this proteins which is run, then you have to look whether the staining process has been done perfectly or not. It means that whether you have done the proper time or proper agitation of the staining of the gel or not. If not, then you have to look into all those steps. And yeah, so that much pretty much about how you can troubleshoot and what are the general different common types of the gel issues which you get in day-to-day protein lab. And for chemicals and reagents, you always have this SAM group which covers most of the basics, how you can prepare the reagents or the chemicals required for the SDS page. So that's it for the troubleshooting.